Hello students, welcome back to Nucleus Neet coaching. I am your Kiran Kumar, Biology Neet faculty. Students, you know that today we are going to start a new chapter, an interesting chapter, Biomolecules. In this chapter we will be discussing about various types of molecules which are found inside the body of living organisms. This chapter is very important. If you understand, if you are thorough with the concepts from this chapter, it will be very easy to understand the three important chapters from NCERT biology, that is respiration in plants, photosynthesis plants, and molecular basis of inheritance. So, in order to understand the three chapters, you should be thorough with the concepts from this chapter, biomolecules. So, get ready with your pen and book. We'll start the class. Students, before understanding what are biomolecules, let us once revise what are molecules. This concept you have already learned in lower classes. However, once again let's revise. Molecules. What are molecules? When more than two atoms are attached with each other by formation of bonds like this. One atom, another atom, and several other atoms are attached to each other, then it results in a structure which is called as molecule. So let us say A is a atom, B is another atom, and a bond is formed between the two atoms. This bond connects the two atoms A and B, then we call this as a molecule. Example, we can take hydrogen. Hydrogen molecule. Hydrogen molecule consists of one hydrogen atom attached to another hydrogen by a bond. It is called hydrogen molecule H2. Another molecule will take water, H2O. In water molecule, we have oxygen attached to two hydrogen atoms. This is water molecule. Another example we will take carbon dioxide. The gas carbon dioxide which is exhaled in our body, carbon dioxide. Carbon dioxide has carbon and it is having two oxygen atoms attached to it. So these are all the molecules. Understood, students? When we break these bonds, what happened? The individual atoms will be released. Understood? So what is difference between a normal molecules like this and our biomolecule? Let's see. What are biomolecules, students? Biomolecules, they are the carbon molecules, means they have carbon atom in the structure, in their structure. They are the carbon molecules found in the cells of living organisms. Where these carbon molecules are present? They are present in the cells of living organisms. These carbon molecules are present in the cells of living organisms. Then we call them as biomolecules. What are the examples of biomolecules? Look here, children. Carbohydrates, proteins, lipids, nucleic acids, hormones, vitamins. So these are some examples of biomolecules which are found inside the cells of living organisms. Understood the concept? Copy down, students. Later we will discuss several examples of these biomolecules. Okay. Okay. First we will discuss about carbohydrates. Carbohydrates are very important biomolecules which forms the main important diet in our day-to-day life. So the food which we eat in day-to-day life, like the rice, chapati, roti, etc. So these food items, they are rich in carbohydrate called as starch. So what is that? Carbohydrate present in rice, chapati, roti. What is that carbohydrate? It is called as starch. Okay. So we can digest this starch, carbohydrate, and we can get energy required for our body. Another example for carbohydrate is glycogen. Glycogen, it is present as a reserve food material in liver, glycogen. Another example is cellulose. Cellulose is the cell wall material in plants. Okay, so these are some carbohydrates. Next, proteins. Proteins also form an important diet in our day-to-day life. So we eat lot of protein-rich food materials such as eggs and meat. Okay, so examples of proteins. The meat which we eat contains myosin protein. What is the protein, children? Myosin, and another protein called actin. So these two proteins are present in the meat such as your mutton and chicken. Myosin and actin are present in the non-vegetarian food, meat. Okay. Casein. Casein is another protein which is present in the milk which we drink every day. Casein. Next, fibroin. Fibroin. Fibroin is a protein which is present in the silk. Silk is produced by silkworm. Okay. So fibroin. Fibroin. Next is keratin. Keratin is another protein which is present in our hair, in our beautiful hair. It is also present in our nails. It is also present in our nails. Keratin protein. Students, all the proteins cannot be digested in our body. Keratin protein cannot be digested in our body. Okay. So, mainly our body can digest myosin, actin, casein proteins. This type of proteins our body can digest to produce energy. Lipids. Lipids also present in our diet. So, the fatty food material which we eat such as the oil, ghee, butter, these are nothing but lipids. They are full of lipid molecules. So, what are the examples of lipids? Oleic acid, linoleic acid. These are the examples of lipids which are found in the edible oils such as groundnut oil, sunflower oil, mustard oil, etc. etc. Understood, students? Next is nucleic acids. Nucleic acids are the acidic molecules which are present inside the nucleus of the cell. Examples are RNA, ribonucleic acid, and DNA, deoxyribonucleic acid. These two are the nucleic acids. Okay, children? Okay. Next, hormones. Hormones are the chemical messengers produced in our body by endocrine glands. Examples you can quote thyroxine. Thyroxine produced by thyroid gland. Next, progesterone, testosterone, etc. These are some examples of hormones produced in our body. Vitamins. So our body can produce some vitamins by itself, but our body requires some essential vitamins from outside through diet. Okay, so these vitamins play very important role in maintaining the life activities inside our body. So deficiency of vitamins will cause several vitamin deficiency diseases. Okay, so what are the examples of vitamins? Vitamin A, vitamin B, vitamin C, vitamin K, etc. These are examples of different vitamins. So these are all the biomolecules. Students, I hope you have got some idea about the concept of biomolecules. Now let us continue. Students, just now we have discussed about different types of biomolecules. Now, suppose if you want to know what are the different types of inorganic elements which are present in the living tissues, then how we can find them? By using Ash analysis test. Ash analysis test will help us to find what are the different types of inorganic elements that are present in the living tissues. So here, in the Ash analysis test, the living tissue is used. The living tissue is first of all dried thoroughly to remove the water, and it is fully burned to obtain ash. The living tissue is burnt at high temperatures. So once burning process is completed, what we get? We get the ash. It is a white, grayish, powdery material. Okay. So once ash is obtained, so during the burning process, all the carbon compounds get oxidized. During the burning process, what happens? All the carbon compounds present in that tissue gets oxidized and gets converted into carbon dioxide gas. During burning process, water vapor and carbon dioxide gets completely removed from the living tissue. Students, I have brought a capsicum vegetable. Okay, so let us consider this is a living tissue because it is full of living cells, isn't it? So this is the living tissue I am taking as an example. So directly I cannot use it for burning. First of all, I should dry this capsicum fruit. Okay, so first of all, I will select my living tissue. I will select my living tissue. So it is my choice. I can either select the animal tissue like muscle tissue or liver tissue, or I can select plant tissue like capsicum or stem tissue or root tissue. That is my wish. Okay, so here I have selected capsicum fruit tissue, which is a plant tissue. Okay, so now what I do is I will dry this capsicum fruit in sunlight. Okay, so once the capsicum fruit gets completely dried, it becomes brown and it will shrink in size. All the water content gets evaporated from this fruit. Okay, so the living tissue, it is dried thoroughly. We can use a sunlight drying method or we can dry inside the oven. So once the living tissue is dried, what happens? All the water content gets removed. Water is removed during drying process. Okay, so once the drying process has completed, our tissue, imagine this is our tissue, living tissue, it will shrink in size. So this is the dry tissue which you have obtained after drying process. Now once the dry tissue is ready, I am going to put this dry tissue in a crucible. So crucible is a small container in which we can burn any type of tissue. Okay, so I am going to put this dry tissue inside this crucible and I am going to provide high temperature of about 450 degrees centigrade. At this high temperature, what happens? My dry tissue starts burning. Literally, it starts burning. So when it starts burning, all the carbon compounds, carbohydrates, proteins, lipids, okay, all this type of biomolecules, they get oxidized and the carbon gets converted into carbon dioxide and it escapes into atmosphere. All the carbon compounds get oxidized to CO2 and it will escape into the atmosphere. Any small amount of water which is left over in this dry tissue, that water also escapes in the form of water vapor. And after complete oxidation, what we get? We will get a white grayish matter that is nothing but ash. It's nothing but ash. Students, after completion of this process, what we get? That tissue, living tissue which we have selected as a capsicum, now it has been converted into a white powdery material called as ash. Now once our ash is ready, now we can analyze the different chemicals which are present in the ash. Okay, on analysis of ash, what we can find? Ash analysis shows presence of inorganic elements. We can find a number of inorganic elements in that white grayish powder. So what are the different types of inorganic elements that can be found in the ash? Calcium, magnesium, sodium, potassium, chlorine, carbonate, sulfate, phosphate, sodium chloride, calcium carbonate. Like this, there will be different types of inorganic elements and compounds which will be present in the ash of living tissue. It can be a plant tissue or it may be animal tissue. Any type of tissue you can find this type of inorganic elements. Understood, students? So this is the technique or the test which is used to find different types of inorganic compounds in a living tissue. Copy down. Students, how can we separate biomolecules from living tissue? We can use a test called acid solubility test. By using this test, we can separate out the biomolecules present in the living tissue, and also we can divide these biomolecules into two groups. That is, the biomolecules with less molecular weight can be separated as biomicromolecules, and biomolecules which have high molecular weight can be separated out as biomacromolecules. Now let us see how we can carry out this test. Acid solubility test. This is the test which is used to separate biomolecules based on molecular weight, based on their molecular weight. So now let us see the procedure, students. Here first of all we need a living tissue from plant or animal. So more preferably we can use the liver from animals. We can use liver tissue from mutton or chicken. Or we can use plant tissue such as cabbage, cauliflower, which are relatively soft and easy to grind. Okay. So now, for now I am taking liver tissue. I am putting a small piece of liver tissue in mortar. So this vessel which is made up of ceramic material is called mortar. I will put liver tissue in the mortar and I will add solvent trichloroacetic acid, CCl3COOH. Trichloroacetic acid. I will add this liquid into this mortar, and once the liquid is added, I start grinding the liver tissue by using a pestle. So when I start grinding the liver tissue by using a pestle like this, what happens? Because of the friction, the liver tissue gets broken, all the liver cells get ruptured, the molecules come out and they dissolve with trichloroacetic acid, and some of the molecules they dissolve in the solvent trichloroacetic acid. So this is the process which is going to happen when we are pestling the liver tissue in the mortar. Okay, so after some time what happens, all the liver tissue gets mixed with trichloroacetic acid and changes into paste like consistency, and finally we get what is called slurry. We get slurry, paste like material. The paste like material of liver tissue, we call it as slurry. Okay, now it is time to filter the slurry. How can we filter the slurry? We are using cheesecloth as a filter. So, or we can use even cotton also as a filter. We put cheesecloth in a funnel. We transfer the slurry from the mortar into this cheesecloth. Okay. Now look here. Once we transfer our slurry, what happens? In the slurry, there is trichloroacetic acid solvent, and there are number of molecules which are already dissolved in the trichloroacetic acid. So the trichloroacetic acid solvent, it starts to drip through the cheesecloth, and this filtrate we are collecting in a beaker. The liquid which is coming out through the cheesecloth we are collecting in a beaker. Okay, once the filtration process is completed, then there is some residual substance left over in cheesecloth. This residual substance which we find in the cheesecloth, it is called retentate. It is called retentate because it is retained inside the cheesecloth. This retentate we can also call it as acid insoluble pool. Acid insoluble pool. Come on students, What is this? Retentate, also called acid insoluble pool. So all those materials which are not soluble in trichloroacetic acid are retained in the cheesecloth as acid insoluble pool. And what about this liquid, clear liquid which is collected in the beaker? The liquid which is collected in the beaker, it is called filtrate. It is called filtrate. The filtrate contains lots of molecules which are able to dissolve in trichloroacetic acid. It is also called acid soluble pool. Acid soluble pool. Understood, students? Now we have separated the two types of materials by using cheesecloth. One is retentate and another is filtrate. Scientists, they analyzed the retentate material and also they analyzed the filtrate material. So once the analysis process has completed, scientists found this thing. So in retentate, scientists found large sized molecules called biomacromolecules. So what did they find in retentate? They found biomacromolecules. Macromolecules, large sized molecules. Biomacromolecules. Why large sized molecules were found in the retentate? Because these molecules couldn't dissolve in trichloroacetic acid. That's why they remained in the cheesecloth in the form of retentate. So what were the different types of biomacromolecules that were detected in the retentate? They detected macromolecules like polysaccharides. Polysaccharides are very big molecules. They have high molecular weight, about 10,000 daltons. Okay, polysaccharides were detected, and they also detected proteins. These molecules are also having high molecular weight, and DNA, RNA, these are nucleic acids. DNA, RNA are nucleic acids. So this type of large sized molecules were found in the retentate, and they found that the molecular weight of these molecules was about 10,000 daltons. 10,000 daltons was the molecular weight of the molecules present in the retentate. Then scientists also analyzed the filtrate. So what they have found in the filtrate, mostly all the molecules were small molecules. Okay, and the molecular weight was between 18 to 800 daltons. 18 to 800 daltons. So in the filtrate, scientists have found that most of the molecules, they had the molecular weight in the range of 18 to 800 daltons only, not beyond 800 daltons, and they called these molecules as biomicromolecules because they are small molecules. Micromolecules. Okay, so what were the different biomicromolecules they detected? So such as amino acids, they are small molecules. Next is sugars, different types of sugars, monosaccharides as well as disaccharides. Sugars, they are sweet in taste. Next, they also detected molecules called nitrogenous bases. These are the molecules which are present in nucleic acids, nitrogenous bases. Next is nucleosides. So these were different small biomolecules which were called as biomicromolecules, which were detected in the filtrate, students. Along with this, some inorganic molecules were also found, such as water, minerals were also detected in the filtrate, that is in acid soluble pool. Then what about lipids? Lipids are also biomolecules, right? So in which fraction they were identified? Actually, lipids are micromolecules, but surprisingly lipids were found in the retentate. What is the reason why scientists found lipid molecules in the retentate? What is the reason? The reason is that the lipid molecule, you can see here, this is a lipid molecule. The lipid molecules have hydrophobic head. This is called hydrophobic head. So because of this hydrophobic heads, they will avoid the water molecules and the solvents. So immediately when they come, the lipid molecules come into contact with trichloroacetic acid or any water medium, these molecules immediately they arrange themselves in the form of spherical ball-like structures which are called as vesicle. What is this structure called as? Vesicle. So in a vesicle, the heads will be facing out and the tails will be inside. So this type of vesicle structure will be formed in the trichloroacetic acid. Once the lipids change into vesicles, they cannot pass through the cheesecloth and they remain trapped in the cheesecloth, and that is the reason you can find lot of lipid vesicles in the retentate. Okay, so actually lipids are biomicromolecules, but where they were found? They were found in the retentate along with biomacromolecules. Okay, so actually we should find lipids in biomicromolecules in the filtrate, but they were found in the retentate because of formation of vesicles. So this is a very important point, students. Understood this concept? Come on, copy down, students. Students, completed copying. I just wanted to share some of my thoughts with you. It has been about 10 years since I have been teaching students NEET biology. I have been teaching since 2014. So many students who took coaching from me, they have settled as doctors in prestigious hospitals, and many students are pursuing medical education in the topmost medical colleges like AIIMS and Zipmer. So what are positive aspects of my teaching? So my teaching will be crystal clear. I will be covering each and every point from NCERT textbook, and I will be explaining the point clearly, and also I will be giving proper notes like this which is legibly and neatly written on the board, and I will be drawing wonderful drawings, diagrams which will be easy for you to understand, and you can always copy this type of diagrams in your notebook. It will be easy for your preparation. Okay, at the end of each chapter I will be discussing some important multiple choice questions which will be asked in the NEET examination. These are several positive aspects which you find in Nucleus Neet coaching. You will not find anywhere else in YouTube. So make sure that you subscribe my channel and also hit the bell icon so that whenever I post a video, you will get the video on your notification, and also share this channel with your friends so that your friends also get benefited. Keep reading, keep happy. All the very best. We will meet in the next class.